Hello everyone and welcome to the episode 2 of Python programming for biology, medicine and genetics. I'm Hamid Reza Bolhassani, PhD of computer engineering, researcher, chief data scientist and founder of DataBiox. I'm very happy to see you again in this episode. As you remember from the previous episode, we are working on this problem, RNA sequence of mouse mammary glands. And we had two data sets. Generally I can say one data set including these 12 samples you see from basal cells and luminal cells for virgin, pregnant, lactating, and for these two cells. Okay, and you remember we have done some operations on this already covered in the previous episode, you can see, and also for better understanding the problem, as I already discussed for you in the previous episode, you can watch my first episode of R programming for biology, medicine and genetics. In that episode I fully explained what is the main problem, but here in these episodes we will cover solving that problem with Python instead of R. Okay, so you remember we had these two, we had two files, yes, this file and another one, and we created the data frame using Pandas. And right now we have a sample info file, yes, sample info, and also a file, data frame name count, yes, here. Okay, so let's see them again. Sample info, this is the dataset sample info. And one thing you can see here, here in sample info, let's go ahead. Here in sample info, it should be the first column name should be sample ID. Let's just change it here, because since we didn't change this unnamed in the previous episode, let's do this because we should correct it, and also as a practice for you. You can right now pause the video and check can you change it or not. And also for reminder, all the data sets, all the information, and also this Google Colab is shared with you in the description. So this is the sample info dot rename columns equal this one, and we will say we wanna change this with sample ID. Okay, let's see what happens. That's great. But if we see sample info again, so why didn't change? Because we should replace. Okay, so let's see again and check. That's great. So this one is correct. We have this sample ID, and then you remember we had this dataset count, here we have this. Also you remember we didn't change that, it should be also gene ID. Let's very fast change it. Count dot rename, and column equal that, that's unnamed, it should be gene ID, yes. So that's right. So we will put this in the count. Okay, and we'll check count. For some of the things that I'm doing here, maybe you find some better solutions. It's very good to tell me or suggest me that a better method as you think. It's very good. I welcome all the comments and feedbacks from your side. Okay, so right now we have this data set. We have this data frame. You see it is a wide, you can see it is a wide data set, and also these samples are in the wide format. We want to make them into line, from wide to line. As you remember in the R practice and in this book that I already shared the link with you, you want to change this wide format to line like this. Okay, because you can see here we have some samples, and count of each sample. You see it's very better presentation for this specific problem. For example, we have this sample and each gene, how many counts of each gene in this sample, and it will help us very good in a better analysis. You can see here is a very better presentation. Here is for each sample how many of this gene is counted. You can see it will help us very much, especially in some problems that you are looking for some reasons for example for a special disease. Which genes are related to a special disease or a special type of cancer or a special grade of a cancer? Okay, so how we can change this format from wide to line? Yeah, so as in the R, which I really love R, it was the syntax. So let's see in Python how we can do this. First of all, we should create this column sample. Okay, so we should first put all these in a column name. Okay, let's try. So first of all, I will create this list column and put a call in the this count data set. Yeah, so I put a for loop here, call in count if call dot starts with, you see the name starts with GSM. You see here the start with GSM name. Yeah, all them are started with GSM. Also in here you can see, so easily we will say GSM. Let's see how it works. Yeah, so I expect that this should be contain all these 12. Yeah, that's right. So the next step, I should create this sequence data, RNA sequence data. Yeah, this data set. So here in R we had pivot longer, but in Python we will use PD. It means pandas. You remember PD was referred to pandas library, which I already introduced here. Import pandas as PD, a very great and a strong library for data science. PD.melt. Yeah, it's a very good method in Python, similar to that one in R for converting the format from wide to line. PD.melt. The name of dataset here, the name of dataset is count, then id vars. You know, you want to keep these two. we want to keep this gene ID and gene symbol. You see, we want to keep these two, so these are ID vars. So gene ID and gene symbol. Then we will have value vars, and this one, the value is the sample column, as you remember, the sample column. Yeah, here is sample column, and var names. Var name, yeah, it also suggests var name is what here? Sample. We want to make this column, the name of this column, sample. You see, the name of this column is sample. Let me, sorry, this is very thick. Yeah, and so this is sample, and also value name. So you also see that here the value name is count. Yeah, with capital C. Here no difference, it's a string, so count. So let's see what will happen. Yeah, that's great. Oh, that's nice. We done it with Python. Yeah, exactly. You can see each of gene ID and the gene symbol. We will see the count of each gene ID in this data set. So one thing that we forgot is to put them in sequence data. Yeah, like this we want to, yeah, this is sequence data. Yeah, 284,820 rows and 4 columns here, sequence data. So I will put this in sequence data. Yeah, and then sequence data dot shape. Yeah, 284,820, yes, 284,000 and 4 column. Yes, and as you remember, it's very clear that it's also two-dimensional. It's very obvious, clear. So we already make it. Yeah, and also you can see that all things work for this. And also, yes, dot describe. Let's see what's the describe. Yeah, okay, but not describe is not usually something useful here, but we can also see the tail, as you remember. Yeah, okay, so we have covered this formatting data, and we have covered how to convert from white to line. Yeah, and we exactly converted the things in R to Python. Okay, so I think for this episode is enough, and you can practice this, and more practice, it will make perfect. And then in the next episode we will try to join in 2 tables, and then go ahead in the future episodes for nice Python visualization, which I really love. Visualization. With visualization we can do a storytelling. We can tell the story of anything. We can extract facts from the data, and I think everything, many things in data science is up to visualization, which give meaning and give life to the data. As you can see, the slogan of Data Bioinformatics is also this: we give life to the data. Okay, it was very nice seeing you again. I hope this episode was useful and informative for you. Kindly share your comments, feedbacks, and anything you wanna tell me. It would be my pleasure. And if you liked it, please suggest this video or my channel to your colleagues and friends who are interested. Hope to see you in the next videos. Bye.